So hello everyone, future doctors and NEET aspirants. So this is another session at our lovely channel PW English Medium, and this is Biswajit, your Zoology mentor at PW English Medium and PW NEET English. Yes, if you are coming from class 11 to class 12 and you have some conceptual gaps while you are preparing for your NEET, now I am here to fill up those gaps in the form of the bridge course session, and the important concepts from the chapters I'll be covering today in this session, that is the two chapters of reproduction and human health and disease. Okay, and if you are watching, you are free to ask your important questions or confusions into the chat box directly. Right, great. So these are the topics will be covered for today's session, and the topics are starting from human reproductive system, the methods of contraception, the science behind infertility and art, right, and common human diseases. Great. So first, let's get started with human reproductive system. Now, if we talk about the reproductive mechanism in human being, NCERT is saying about some events. So what are those important events in the course of human reproduction? So starting from gametogenesis. Gametogenesis. What do you mean by gametogenesis? The process of production of male and female gamete, that is called gametogenesis. Gametogenesis takes place inside the primary sex organ or inside the gonads. For male, there is one pair of testes. For female, there is one pair of ovaries. Yes. So the whole process of gametogenesis we need to study. The second phase, what after formation of gamete, is the male partner who will be transferring the male gametes into the reproductive tract of female through the process of insemination. So after gametogenesis, the second most important thing, that is insemination, the process of transfer of male gametes or sperm into the female reproductive tract. Next, what after insemination? Now the sperms in the form of a semen will get inside the reproductive tract of female and will reach into the ampulla or ampullary isthmus region, and it will wait for the secondary oocyte or immature ovum, right? Now what if simultaneously, if secondary oocyte and the sperms are present into the ampullary isthmus region of female reproductive system, the sperm will be chosen by the secondary oocyte for the process of fertilization. So third phase, that is fertilization, the fusion of male and female gamete. What after fertilization? After fertilization, there will be formation of the single diploid cell, that is called zygote, and gradually the zygote will start embryonic development from 1 cell to 2 cell to 4, 4 to 8, 8 to 16, and gradually a special stage of embryo will get implanted into the innermost wall of uterus or mother's womb, and the process and the stage is called as implantation. Okay, now you need to remember here which stage of embryo is getting implanted into the inner wall of uterus, and the stage is blastocyst stage of embryo, a football like hollow stage. Now what after implantation? That implantation announces the good news, that is the starting of gestation period or pregnancy. So the fifth stage, that is gestation, and it will stay for 9 months in human being, and after 9 months it's time for childbirth or delivery of the baby, and that phenomena is called as parturition. So these are the sequential events you will be finding while you are studying for studying human reproduction. Yes, Mr. Maran, very happy evening and welcome to the class. Great. So let's talk about male reproductive system first. Okay. So whenever we talk about reproductive system and their anatomy, that is structure, we need to talk about the structure and their functions. Okay. So for male reproductive system, there are primary sex organs, primary sex organ or primary reproductive organ, and secondary sex organs or secondary reproductive organs. Now, what about primary reproductive organ or sex organ in male? That is one pair of testes, two testes. They are called as primary sex organ. And what is the function of these two testes? They are responsible for sperm production, and the process is known as spermatogenesis. And these testes, the special cells inside the testes, they are responsible for production of the male sex hormone called as, you know it as testosterone, and we call it as androgens. So androgen production is also one very important function of male reproductive system. Yes, Avilasha, very good evening, welcome to the class. So what are the functions of primary sex organ or primary reproductive organ? You can call these as gonads, right? So function of gonads or primary sex organ for male testis, and the function is spermatogenesis, means sperm formation, and secondly production and secretion of male sex hormones, and we call them androgens. And the testosterone is the prime androgen, the main androgen produced inside human male testes. Okay. Now, what about secondary sex organs? Who are they? There are some ducts, some glands. Secondary sex organ includes some ducts, accessory glands, and external genitalia. Now, if you ask me, sir, what is the function of these secondary sex organs? They are responsible for providing nourishment, nutrition to the sperms. So that is the main function of secondary sex organs, and also they help us to transport the sperm, right? So transportation of gametes and nourishment of gametes are the functions, major functions of secondary sex organ. You need to remember that secondary sex organs are not responsible for production of gametes, or even they cannot produce sex hormones or any hormones. Now, what are the ducts present inside the male reproductive system or accessory ducts? Okay, as given in NCERT, they are seminiferous, starting from inside testis, you will be finding seminiferous tubule, right? And after that, when it's leaving the testis, starting from retitestis, then vasa efferentia, epididymis, vas deferens, ejaculatory duct, and urethra or urinogenital ducts. So what are they? Sperms are getting produced with the process of spermatogenesis inside the seminiferous tubules of testis, and when they are released from seminiferous tubule through this passage, through retitestis, vas efferentia, epididymis, vas deferens and ejaculatory duct, they are going to come out through urethra, and the opening will be calling it as urethral meatus or urethral opening. Okay. Now, what are the specific glands you will be finding inside male reproductive system? The glands are one pair of seminal vesicles, one prostate gland, and two Cowper's gland. So, three types of glands are present: one pair of seminal vesicles, one pair of Cowper's gland. You can call this Cowper's gland as bulbourethral gland, and one single prostate gland. So, every gland is secreting some very crucial component which is responsible for the formation of semen. Now, what is semen? Yes, namaste Ayush. Now, what is semen? Semen is a mixture of male gamete or spermatozoa and a liquid which is filled with nutrition enzymes, right? Now, this nutritious fluid called as seminal plasma, that is secreted by seminal vesicle, prostate gland and Cowper's gland. Now, this seminal plasma along with the sperms or spermatozoa, that is called as semen. Okay. Now, what are external genitalia? External genitalia means the parts of male reproductive system which is visible from outside, and we call it as external genitalia, and they are one penis and one scrotum. One penis and one scrotum, they are part of external genitalia, and inside that scrotum, it's a skinny sac, you will be finding two scrotal sacs are present. So, this is a very brief table or chart of male reproductive system. Now, first we will be talking about the primary reproductive structure of male, that is testis. So, let us talk about testis first. Okay, before going to testis, a brief diagram of male reproductive system I will be drawing. So, this is scrotal sac and this is testis, which is divided into testicular lobules. Inside each testis you will be finding 250 chambers are there. Outside the testis you will be finding a supercoil structure called epididymis, and this epididymis from the tail region will give rise the sperm duct, or you call it vas deferens, which will move towards the abdominal cavity and will make a U-turn or looping near to the urinary bladder. This is the urinary bladder, and that loop structure, even we call it the vas deferens, will mix with seminal vesicle. The duct from seminal vesicles which are coming, we call it seminal duct. So, seminal duct along with the sperm duct or the vas deferens will merge together to form ejaculatory duct, right. So, this particular portion is called as ejaculatory duct, and how it is formed? The merging of seminal duct along with the vas deferens. Okay. From where this vas deferens is taking the looping pattern or U-turn? At the junction of the ureters and the urinary bladder. You need to remember this particular statement. At the junction of ureters and urinary bladder, this vas deferens will take the U-turn and again it will descend downwards and it will merge with two seminal duct to form ejaculatory duct. Okay. Now from here, from the urinary bladder, urinary urethra is coming down. Now this urinary urethra will merge with the ejaculatory duct. Here the prostatic duct will also join with them to form a common urethra, and this common urethra or urinogenital duct will come outside through penis and the opening that is called as urethral meatus. Okay. Now what about Cowper's gland? Already we got these seminal vesicles, two seminal vesicles, one prostate gland, right? Now here two smaller glands are present. We call them Cowper's gland or bulbourethral gland. So their duct along with alkaline mucus that will also open into the penile urethra, and that will the secretion of these bulbourethral glands will help to maintain the viscosity or the fluidity nature. It will make the urethral lumen alkaline, neutralizing the acidity of the urine, because this is the same passage through which urine also will come out and semen also will come out. Now semen contains sperm and acidity is very much toxic for spermatozoa. That's why the passage you need to maintain the neutrality. You need to maintain the basicity. The more basic the environment is, the sperms are healthy. If the environment is acidic, then sperm will start dying. Yes. Hello Sahana, very good evening. Okay, Chetu, hello. So that was the important structure of male reproductive system. So this is scrotum, this is testis, this is epididymis, this is vas deferens. Okay, this is ureters which are joining with this urinary bladder, and from here only this vas deferens will take a loop and will come down, and these are seminal vesicles. These are seminal ducts. The ducts which are coming from seminal vesicles, they are called seminal ducts, and along with this vas deferens they will form ejaculatory duct. Now ejaculatory duct along with the urethra coming from the urinary bladder, urethra coming from the prostate gland, this is the prostate, will make the common urethra or urinogenital duct. Urinogenital duct, or simply you can call it as urethra, right? Okay, and these little glands here, they are the Cowper's gland or bulbourethral gland, right? Okay, so now we'll move forward to the detailed discussion of each testis. Now as already I was saying that testis is called as a gonad or primary reproductive organ, and they are situated outside the abdominal cavity. This is the abdomen. This is the abdominal cavity, but one pair of testes there situated outside the abdomen, inside the scrotal sac. So these testes are also called as extra abdominal organs. extra abdominal organ. Why extra abdominal? Because they are present outside the abdomen. But during the time of embryonic development, if you want to track down the history of the position of the testes, then you will not be finding the testes are present inside the scrotum during the time of embryonic development, specifically before the 7th month of embryonic phase, right? So during the time of embryonic development, you will be finding this testis, one pair of testes, they are present inside the abdominal cavity, and with the help of some cord like structure called as spermatic cord, gradually during the time of morphogenetic development or embryonic development, this one pair of testes will descend down and will get fixed inside that scrotum, right? So you need to remember one more terminology here, the cord, which is telling us about the previous position of testes inside the abdominal cavity. That cord is called as spermatic cord. That cord is called as spermatic cord. Now I will talk about one disease, and the name of the disease is cryptorchidism. 0.0001% case, means rarely in case of males you will be finding that scrotums are empty but testes are present inside the abdominal cavity. So in that case what will happen, that male will not be able to produce sperm, because for spermatogenesis you need to maintain a special temperature, that is 2 to 2.5 degree minus than your body temperature, right, which is not possible inside the abdominal cavity but possible into the scrotal sac. So that particular condition is called as cryptorchidism. That particular condition is called as cryptorchidism, when testis fails to descend into the scrotal sac during the time of embryonic development. So in case of cryptorchidism, scrotums will be empty but testes are present into the abdominal cavity, right? Okay, so let's talk about the anatomical aspect of testis. If you take a dissected testis, then you will be finding the testis is having some chambers inside, and these chambers are called as testicular lobules. These chambers are called as testicular lobules, and the covering of testis, that is called as tunic. The covering of testis, that is called as tunic. This is the outer skin or the scrotal skin. Now below the scrotal skin, the testicular covering is present, that is called as tunica vaginalis. The first layering of testis or the first covering of testis, that is called as tunica vaginalis. Next, after that, the main covering or the second covering of testis, that is called as tunica albuginea. Tunica albuginea. Why that is so much important for us, this tunica albuginea layer? While a male is running or jumping, there will be chance of friction or production of shock that can damage the testis. So it's the liquid present into the tunica vaginalis and the thick outer covering called tunica albuginea, fibrous covering, which is protecting the testis from all kind of shocks and friction. So thank God males have tunica albuginea covering outside the testis, and it's the tunica albuginea which is responsible for formation of these compartments inside the testis, and these compartments are called as testicular lobules. These compartments are called as testicular lobules. And how many such lobes are there? There are 250 compartments are present inside the testis, right? And if you check each compartment, the inner lining is made by another layer that is called as tunica vasculosa. That is called as tunica vasculosa. So, there are total 3 coverings of testis are present. Outermost covering is tunica vaginalis, but the most important covering, that is called as tunica albuginea, right? And this tunica albuginea is forming these different chambers, and the innermost layering of testis, that is called as tunica vasculosa, because that is highly vascular with full of blood supply. Now, what is present inside one single chamber? Inside one single chamber, you will be finding highly coiled tubes which are responsible for production of sperm. The sperm production factory, these highly coiled tubes are called as sperm production factory, and we call them as seminiferous tubules, right? So, inside each chamber, these highly coiled tubes are present. They are known as seminiferous tubule. Seminiferous tubule. They are responsible for sperm production, and they are tiny coiled tubes. That's why they are called as tubules. Tiny tubes are called as tubules. And you have to remember, seminiferous tubules are responsible for what? Spermatogenesis, sperm production. Spermatogenesis is taking place inside the seminiferous tubules. Now, inside one chamber, how many seminiferous tubules are present? Inside one chamber, one seminiferous tubule can be present. 2 seminiferous tubules also can present. You will find some chambers with 3 seminiferous tubules inside, right? So, overall average 2 seminiferous tubules are present into each chamber, and one testis is containing near about 500 to 700 seminiferous tubules continuously who are producing sperms. Okay. Now, these seminiferous tubules will be coming outside and will join together to form this network-like structure, and that is called as retitestis. That is called as retitestis. Why it's named as retitestis? Because that's making a network, reticulate structure, right? Reti means which is like network. Now, from retitestis, few more tubes will go outwards, and these tubes are called as vasa efferentia. Why they are called as vasa efferentia? Because these tubes or these vessels, they are bringing these sperms outside, outwards. They are bringing sperms outwards. That's why they are called as vasa efferentia. Okay. Now, all those vasa efferentia will join together. This 15 to 20 vasa efferentia will join together, and they will form another highly coiled structure, and this highly coiled structure is called as epididymis. This highly coiled structure is called as epididymis. Now, how this epididymis is formed? By the when this vasa efferentia will join together and will form highly coiled structure outside the testis, and that highly coiled structure is called as epididymis. This epididymis is having 3 segments. This part is acting like a cap of epididymis or head of epididymis. You can call it caput epididymis. This part is the body of epididymis or corpus of epididymis. This end part is the tail of epididymis or cauda epididymis. So, epididymis is having 3 part: head, body and tail, and you need to remember this tail is responsible for, this tail region is the region for origin of sperm duct or vas deferens. So, again I am repeating it. Inside the testis there are some chambers. Those chambers are called as testicular lobules. inside one testis 250 testicular lobules are present, right? Okay. Inside one testicular lobule or inside one chamber, these tiny coil tubes are present. They are called as seminiferous tubules. Total inside the testis 500 to 700 seminiferous tubules are present, and inside one chamber 1, 2 or 3 seminiferous tubules can be accommodated. When these seminiferous tubules are coming outside, they will join together to form a network-like kind of structure, and that network is called as retitestis. From this network, you will find that some tubes are coming outwards. They are called as vasa efferentia. This 15-20 vasa efferentia will join together to form another highly coiled tube, and that is called as epididymis. Now this epididymis has 3 segments. The upper part is called head of epididymis or caput epididymis. The middle part is called as body of epididymis or corpus epididymis. The lower part of epididymis is called as tail of epididymis or cauda epididymis. And this tail part is very much crucial. Why? Because it's the tail of epididymis from which vas deferens will merge out. This vas deferens is originated from the tail or the cauda part of epididymis. So this was the basic anatomical structure of testis. Now we will get inside one testicular chamber to check what is present there. Okay. Suppose you have taken one testis, these are the chambers. Inside the chamber, these are seminiferous tubules. Now you are zooming it out. You are zooming it out to check what are they actually. Okay. First, you will find the cross section of seminiferous tubule. This is the cross section of seminiferous tubule. Inside this seminiferous tubule, you will be finding some tall columnar cells, tall elongated columnar cells. These are called as Sertoli cell, and some oval to cuboid shaped cells. These are called as Ugonial cells. So inside the seminiferous, this tube is the cross section of one seminiferous tubule, right? Okay. So inside the seminiferous tubule, what you are gonna get? These cells, this spherical or cube shaped cells, they are the Ugonia. Sometime in the initial form, in immature testis, we call them primordial germ cells, right? In initial testis, these Ugonia are called as germ cells because these are responsible for production of sperm later on. So that's why initial form, this Ugonia, we call them as germ cell. They are diploid in nature. Now these tall slender cells, they are called as, these tall slender cells are called as Sertoli cell. They are also diploid in nature. Sertoli cells are also diploid in nature. What is the function of Sertoli cell? Sertoli cell will be producing some important factors which are responsible for maturation of sperm and providing nutrition to the spermatogenesis process. While the Ugonia is undergoing spermatogenesis, so the Sertoli cell will be providing nourishment to those cells, to those developing cells, and will secrete some hormones, some factors which are mandatory for the ultimate maturation of spermatozoa. Yes, Anamika, very good evening. Okay, so that was the thing present inside the seminiferous tubule. This is one seminiferous tubule. Now, the space which is present outside the seminiferous tubule, the space outside the seminiferous tubule, that is called as interstitial space. That is called as interstitial space. The space outside the chamber, that is called as interstitial space. And what are the things you are gonna get inside this interstitial space? Okay, this interstitial space has some very important cells. Without these cells, sperm formation is not possible. Without the secretion of these cells, because we call them as Leydig cells. We call them as Leydig cells. Sometime you call it as interstitial cell, interstitial cells or Leydig cell. So another name of Leydig cell is interstitial cells. Okay. Now, why they are so much important? Because it's the Leydig cells who are responsible for secretion of the male sex hormone called androgens. You know it as testosterone. So, Leydig cells or interstitial cells are responsible for production of androgens, right? And these androgens are necessary for the development of male reproductive system, and also these androgens are responsible or necessary for the formation of sperm. Okay. Apart from Leydig cell, you will be finding some blood vessel, some tiny small blood vessels. Apart from Leydig cells, you are gonna get some small blood vessels into the interstitial space. Apart from Leydig cell and small blood vessels, you will be finding some WBC. You will be finding some WBC as well, which are called as immunologically competent cells. Immunologically competent cells, they are nothing but WBC roaming inside the interstitial space of testes, and they are responsible for this the immunologically competent cells or these roaming WBCs inside the interstitial space. They are killing all the germs present into the interstitial space, or they are clearing the dirt particles from here. But remember, these immunologically competent cells, they cannot take entry inside the seminiferous tubules, and that is taken care by the Sertoli cells, that is called as blood testis barrier. That is called as blood testis barrier. Okay. So, that was more or less a brief about this particular slide. Okay, now Utkarsh is asking, can a student preparing from another competitive exam buy this batch Lakshya NEET if student bought by mistaken, even if their goal is not NEET? My goal is CUET or government university entrance, help. See Utkarsh, your question actually it's a good question, right? And my answer will be yes, if you are preparing for NEET, CUET will be much easier for you to clear. More or less the syllabus is similar. Listen Utkarsh carefully. The syllabus for CUET and NEET, that is similar, more or less it is similar, but the difficulty level of NEET is much higher than the NEET examination, than the CUET examination. NEET is tougher, CUET is little bit easier, right? So, if you are, suppose I am giving you example in comparing with other example, if you are a batsman, if you are a cricketer and you are playing for your country, you are a player of national team, so will it be harder for you to play for a state level team? It will be easier, right? So, that is the charisma. So, by mistake or whatever you are saying, if you bought already the batch called the Lakshya NEET English, right, stick together with the quality content, right? Each and every aspect of NEET as well as CUET also will be covered. So, do not get panicked, my dear. Got it? Okay. Okay, so we are done with male reproductive system, the basic structure of male reproductive system, the basic structure of testes. Now we will talk about the external genitalia. Okay, external genitalia of male. That means scrotum and penis. Okay, so scrotum means a skinny sac-like structure. A skinny sac-like structure is called scrotum, and the scrotal septia, septum, is dividing the sac into two chambers. So each scrotum is having two scrotal sacs, and inside the scrotal sac one of the testis, each testis is placed, right. And the muscles of scrotum, will be helping us to maintain the position of the testis depending upon the season, right. You will be observing during the time of summer the muscles are loosened, relaxed. During the time of winter the muscles are contracted. In that time the scrotal sacs are very tight, you feel very tight. So this behavior is called adjustment or accommodating the position of testis depending upon the body temperature, because you know the process of spermatogenesis needs -2 to 2.5 degrees Celsius in compared to our body temperature, which is 37 degree. So in 37 degrees Celsius temperature, sperm production is not possible at all. That temperature will not induce the proliferation or the differentiation of those germ cells. That is why the temperature is very much significant, right. Okay. Now penis is also called as copulatory organ. Why penis is called as copulatory organ? What is the meaning of copulation? The course or the process, the mechanism of intercourse, or you can call it coitus. And why coitus is necessary? Because it's the coitus through which during the time of coitus a male partner is transferring the semen into the female reproductive tract, and that is why we call this structure as copulatory organ. Now if you observe it carefully, if you observe the penis carefully, it is covered by a retractile skin that is called as foreskin. The foreskin, that is a retractile skin which is partially covering the penis, right. The shaft of penis, the skinny spongy end, that is called as glans of penis or glans penis. The pipe which is coming through the penis, that is called as urethra, right, and the opening is the urethral meatus or urethral opening, or you can call it urethral meatus, and through this urethral meatus male will be ejaculating semen as well as passing the urine. That is why this is also called as urinogenital duct, right. Okay. Now a male can understand why whenever a male is getting sensation, right, or increasing blood pressure due to the result of increased blood pressure towards the penis, there will be a phenomena caused with exhibited by the penis, that is called as erection. So the erection of penis is due to presence of some special muscles. So they are called as erectile muscles. Now who are they? The most important erectile muscles, they are present towards the dorsal side of penis. So this particular muscle which is present toward the dorsal side of penis we call it as corpora cavernosa. Corpora cavernosa. So it will produce hydraulic pressure. Basically, penis is a particular organ which doesn't have any bone inside it, but still it erects due to the presence of corpora cavernosa and the hydraulic pressure and the blood pressure produced by it, right? Okay. Another tissue is surrounding this urethra, and the tissue which is surrounding the urethra and the tissue which is responsible for formation of these glands of penis, that is called as corpus spongiosum. Corpus spongiosum. So, together the corpora cavernosa and corpus spongiosum, we call them erectile muscle, and they are responsible for erection of penis, right? And few MCQ questions are also asked in PYQs, like urethral meatus is through which of the following tissue? So, you have to answer urethra or urethral opening is coming through corpus spongiosum tissue, right? Glans penis is modified portion of which section? Glans penis is modified portion of corpus spongiosum, not it is not modification of corpora cavernosa. Glans penis is a modification of corpus spongiosum only. Now, we will talk about some accessory glands of human reproductive system. And we know there are three kinds of glands are there. First, seminal vesicles. Second, prostate. And thirdly, bulbourethral gland or Cowper's gland. So, these three kind of glands are present inside male reproductive system, and you know the number: seminal vesicle in a pair, prostate single, Cowper's gland, they are also present in a pair. Okay. The secretion of seminal vesicle is called as seminal fluid. The secretion of prostate is called as prostatic fluid. The secretion of Cowper's gland is called as alkaline mucus. And all together, seminal fluid plus prostatic fluid plus alkaline mucus, that is called as seminal plasma. That is called as seminal plasma. So, whenever you will see this particular word seminal plasma, it is very usual that the term that will click your mind, that is, this is the secretion of seminal vesicles, but that is a wrong concept. Seminal plasma is the overall secretion of seminal vesicles, prostate and bulbourethral gland, right. So, what is the secretion of seminal vesicle individually? The name is seminal fluid, right. Okay. Now, seminal fluid is alkaline in nature. Cowper's gland fluid, that is alkaline mucus, that is also alkaline in nature, but the secretion of prostate gland, that is prostatic fluid, that is acidic in nature. You need to remember this. Secretion of seminal vesicle and bulbourethral gland, they are alkaline in nature, but secretion of prostatic fluid, that is acidic in nature. Some books it is written that the secretion of prostate gland is also alkaline, but that is a wrong concept actually. That condition is called as prostatitis, but in normal condition the prostatic fluid will be acidic, but rest of the things will be alkaline in nature. So, what will be the overall alkalinity of semen or seminal plasma? Overall alkalinity will be basic or alkaline, because 70% volume of this seminal plasma will come from seminal vesicles only, right. And NCERT is writing about seminal plasma only. It is rich with fructose. Seminal plasma is rich with fructose and some enzymes. Okay. Now, why this fructose is much important? The fructose is the primary food or source of energy present into the semen, which is necessary for spermatozoa, right. how this spermatozoa will swim. They need energy, mitochondria present inside them, but they also need uh, energy. So from where they will be harvesting energy? When they will obtain energy source, and this fructose will act as energy source for them. Now if you are asked, instead of fructose, can glucose be used for the energy source for spermatozoa? In artificial condition, in laboratory condition, yes, instead of fructose, glucose can be used as the energy source of spermatozoa. But in our body, when a male body is producing semen, uh, inside seminal vesicles, they are releasing fructose only. There will be no production of glucose or no secretion of glucose inside the seminal plasma, right? So moving forward to the next thing. Now what is semen? So semen is simply the combination of sperm or spermatozoa plus seminal plasma. Okay. Now, during the time of one ejaculation, a mature healthy male can release or ejaculate 2 to 4 ml of semen, and that contains nearly 200 to 300 million spermatozoa. Right? You need to remember this particular information. One ejaculum of a mature male contains 200 to 300 millions of sperm. Okay. Now, out of this 200 to 300 millions of sperms, 60% sperm should have normal shape and size, and out of this 60%, 40% should pose vigorous motility. Vigorous motility should be present to the 40% out of the 60% spermatozoa. What do you mean by vigorous motility? The capability of moving very fast. So it's only the first moving sperms or energetic sperms will create a possibility to fertilize the secondary oocyte, right, to produce diploid zygote. Now we'll talk about female reproductive system. So like male reproductive system, females, they also have primary reproductive organ, and as primary reproductive organ, they have one pair of ovaries. What are the functions of ovaries? Production of ovum and secretion of hormones like estrogens, production of hormones like progesterone. So estrogens and progesterone, they are the prime hormones produced by ovaries. They are also called as female sex hormone. So that's why ovaries are also called as gonads in case of female. Now let's come to that section when while we will be discussing about the secondary reproductive structures. If you talk about the secondary reproductive structures in case of female, you will be finding some accessory ducts, you will be finding some accessory glands, you will be finding some external genitalia as well. Okay. Now, which are the organs present as ducts, accessory ducts in female reproductive system? From outside to inside, it's vagina, uterus, cervix, uterus, and fallopian tube. The glands of female reproductive system, they are called as Bartholin's gland, glands, and paraurethral gland. Paraurethral glands. So these two kinds of glands are present inside the female reproductive system, and the parts of external genitalia of female. External genitalia is also called as vulva. External genitalia of female reproductive system is also called as vulva, and that contains mons pubis. Mons pubis is a kind of fatty cushion. Okay. Skinny fold, that is called as labia majora. Labia majora. From labia majora, there will be origin of labia minora. After labia minora, at the upper junction of labia minora, you will be finding clitoris. Then the membranous girding of vaginal opening, that is called as hymen. So mons pubis, labia majora, labia minora, clitoris, and hymen, these are some important parts of external genitalia. Now let's move forward to the detailed diagrammatic representation of female reproductive system. Okay. So this is female reproductive system, the diagram of female reproductive system. These two are ovaries. These ovaries are attached to the uterus as well as the abdomen with some ligaments. This inverted pear-shaped structure, that is called as uterus. The upper part of uterus is called as uterine fundus. This cavity is called as uterine cavity. And inside this cavity, the implantation will take place, and the layer, if we talk about the layers, there are three layers. You will be finding the innermost layer, that is called as endometrium, outermost layer perimetrium, and the middle layer, that is myometrium. So implantation of embryo and the periodic menstrual changes, all of those changes will be taking place in with this structure called uterine endometrium, right? Okay. And towards the vagina, this is the vagina, a non-glandular part, and the narrow passage in between the uterine cavity and vagina, that is called as cervical canal. Cervical canal. and this whole region is called as cervix. Whole region is called as cervix, and this arm like structure, these are called as fallopian tube. There are some other names of fallopian tubes you need to remember. So what are those names? You can call it as oviduct. Another name of fallopian tube, that is uterine tube. Uterine tube is another name of fallopian tube. So fallopian tube got three names. One is fallopian tube, another is oviduct, another is uterine tube. Now this funnel like structure, that is called as infundibulum. Infundibulum. The finger like projection along with this funnel, these structures are called as fimbri. The narrow widest part of fallopian tube, that is called as ampulla, and according to NCERT, ampulla is the site of fertilization, and the narrow part which is joined with the uterine cavity, that is called the isthmus. So there are three parts of fallopian tube, starting with infundibulum, then ampulla, then isthmus. The finger like projections of infundibulum, that is called fimbri. What is the function of infundibulum? The infundibulum is containing a narrow opening called as ostium. With the help of ostium, after ovulation, the secondary oocyte will be collected by this infundibulum. Okay. Now this is the vaginal opening, and sometimes this vaginal opening is partially guarded by a membranous structure called hymen. Vaginal opening is never completely guarded by hymen. So assume if vaginal opening is completely guarded by hymen, then what will happen? After the process of menstrual bleeding, the menstrual blood will not get the outer passage. That's why if vaginal opening is guarded by, if you are finding that vaginal opening is guarded by hymen, but still it will be porous, it will be partially guarding the opening or vaginal meatus. Now we will talk about ovary. So if you enlarge the diagram and try to observe the ovary, one single ovary, the outermost layer of the ovary is called as ovarian surface epithelium. Ovarian surface epithelium. Below ovarian surface epithelium, we will be finding a layer of fibrous tissue called as tunica albuginea. Tunica albuginea is present below ovarious surface epithelium, and the inner space of ovary is called as ovarian stroma. The inner space is called as ovarian stroma. So who is covering ovarian stroma immediately? The immediate covering of ovarian stroma is tunica albuginea. And which layer of epithelial tissue is covering tunica albuginea? Ovarian surface epithelium. They will be containing some cuboidal cells. Now let us check the ovarian stroma part. The innermost part of ovarian stroma, that is called as medulla. The outer part of ovarian stroma, that is called as cortex. Like other organ, the superficial part is called cortex, the inner part is called as medulla. What is the importance of medulla? Medulla is the entry point of the nerves and blood vessels. Medulla is the entry point of nerves and blood vessel inside the ovary. And in cortex, you will be finding developing phases of follicles and developing phases of egg. So, developing follicles or eggs are present into this cortex region, and entry point of blood vessel and nerves, this medulla zone is representing rich blood vascular supply and the nerve supply zone. So, that was the overall anatomy of ovary. Now, we will talk about gametogenesis, the process of sperm and ovum production. So, the first step of sperm production will start from spermatogonia. So, you can choose any random male. Inside the testis, the cells are present. We will be calling them as spermatogonia, all the germ cells. Okay. Now, at the age of puberty, means at the age of 12 to 13 years of age, gradually what will happen? This spermatogonia cells will start division, start proliferation. So, the spermatogonia will produce more spermatogonia. A similar kind of cells are produced. So, which cell division is required to produce similar kind of cells? That is mitosis. So, spermatogonia will produce more spermatogonia cells by the process of mitosis. Now, out of these spermatogonial cells, few spermatogonial cells will undergo differentiation. They will gradually change their shape and we will get a new kind of cell. They are called as primary spermatocyte. Now, the real story will start from here. This primary spermatocyte will start first meiotic division. Now, they will not undergo, they will not develop by the process called mitosis. Now, they will start meiosis division. Now, what is the difference between mitosis and meiosis? Resultant cells of mitosis are always diploid in nature. They have similar number of chromosomes in compared to their mother cell. But for meiosis, there are two phases, first meiotic division and second meiotic division. For first meiotic division, one particular diploid cell will divide into two daughter cells and those two daughter cells will have only half number of chromosomes, right. So, the main difference between mitosis and meiosis is, for mitosis chromosome number will not change, but in case of meiosis chromosome number will reduce to half. So, that is why meiosis is also called as reductional division and mitosis is called as equational division. Now, this is the first step of meiotic division, M1 or first meiotic division. What will happen? This primary spermatocyte will divide to produce secondary spermatocyte. Now, what will happen with secondary spermatocyte? I was discussing there are two phases of meiosis, only meiosis 1 is completed. Now, meiosis 2 will start and this secondary spermatocyte will get modified into spermatids. So, gradually the cells are changing their nature. Starting from spermatogonia, they are diploid cells. from few spermatogonia to many spermatogonia. So, this spermatogonia means they are also diploid in nature. Now, few of this spermatogonia cell will undergo differentiation. Differentiation is not a kind of cell division. It's that some cells are changing their shape, changing their nature, and they will form a new kind of cell called as primary spermatocyte, but they are also diploid. But as soon as they have completed first meiotic division, will get secondary spermatocytes, they will become haploid, chromosome number will be reduced. Now, from secondary spermatocyte, as a result of second meiotic division, spermatids are formed and they are also haploid in nature, because meiosis 2 will not reduce the number of chromosome. So, if you are asked which is the first haploid cell in the life of developmental life of sperm, so you need to answer first haploid cell, that is secondary spermatocyte. If you are asked which cell will undergo first meiotic division, you need to answer primary spermatocyte cells will undergo first meiotic division. Second meiotic division, secondary spermatocyte, and resultant cells of second meiotic division, they are spermatids. Now, cell division is completed here. Now, with some help, now we need some help from Sertoli cell. Remember the tall elongated cells present inside the seminiferous tubules. Now, with the help of Sertoli cell, they will send some signal in the form of sperm maturation factor. Now, with the help of sperm maturation factor which is coming from Sertoli cell, these spermatids will get modified into sperms or spermatozoa. So, the sperms or spermatozoa, they are the structural matured form of sperm. Spermatozoa are the structural matured form of sperm, and this last step, the formation of spermatozoa from spermatid by the influence of sperm maturation factor, this is a differentiation phase. This is a differentiation, and we call this as spermiogenesis. Spermiogenesis. Spermiogenesis is not a kind of cell division, rather it is a kind of differentiation. So, the whole process from spermatogonia to spermatozoa that is called as spermatogenesis. That is called as spermatogenesis. And the last, only the last step of spermatogenesis, that is called as spermiogenesis, and everything will start just after the puberty, the age of 12 to 13 years. Okay, now we will talk about oogenesis, the process of ovum formation. The oogenesis will start with oogonia, but unlike spermatogenesis, the starting phase of oogenesis is embryonic life of the female. So, during the time of embryonic life of a female, this oogonia will divide and will differentiate to form primary oocyte. Now, how oogonia reached into primary oocyte stage? By the cell division mode called mitosis as well as differentiation. Okay. Now, how many primary oocytes are produced inside one ovum? Millions, 2 to 4 millions of primary oocytes are produced inside each ovary, and during the time of embryonic phase only, this primary oocyte will start meiosis 1 or first meiotic division, but it will get arrested at the prophase 1 phase, prophase 1, particularly at the diploteny phase of prophase 1 of meiosis 1, that will get arrested. Means the cell division will stop. Arrest means cell division is not getting a green signal, rather it's getting a red signal from your body system. And all these steps are taking place during the time of embryonic life. Okay. Now the sequential phenomena, birth will be there. After birth, puberty will arise. Now, upon arrival of puberty, female body will start producing two very important hormones from anterior pituitary, and they are FSH and LH. So, these two hormones are very much necessary in order to resume the process of meiosis 1. So, what will happen after puberty? Meiosis 1 will be completed, and this primary oocyte will divide to form two cells. One is called as secondary oocyte, and another is called as first polar body. First polar body. This first polar body will die within few hours, and this secondary oocyte will be ovulated. The secondary oocyte will be ovulated from ovary. Now, again this secondary oocyte will start second meiotic division, but again it will get arrested. Again it will get arrested because it's lacking some important factors to complete meiosis 2. Now, in which stage of meiosis 2? Second time this secondary oocyte will be arrested at metaphase 2 stage. At metaphase 2 stage of meiosis 2 or second meiotic division, the secondary oocyte will initiate second meiotic division, but will be stopped. Okay. Now, is there any probability this something will come with a gift and this meiosis 2 will be resumed or continued again? Yes, there is only one possibility, that is arrival of spermatozoa. If fertilization is taking place, then only it is possible that this meiosis 2 will be completed and the secondary oocyte will develop to form one mature ovum or oocyte along with a smaller second polar body. As usual, the second polar body will die within few hours, and this oocyte is performing fertilization process with the spermatozoa and will get converted into zygote within some subsequent within few hours. So, that was whole idea about the oogenesis. So, what is the basic difference? Spermatogenesis will start at the age of 12-13, that is after puberty. And for the whole and soul process of spermatogenesis, from the whole and soul process of spermatogenesis, you can observe differentiation is taking place twice. Once when spermatogonia are getting converted into primary spermatocyte, and secondly when spermiogenesis is taking place. So, for the process of spermatogenesis, twice we can observe differentiation. Rather, in contrast to the process of oogenesis, the differentiation will take place only once when these oogonia cells are getting converted into primary oocyte. The second time there will be no differentiation, as because oocyte is considered as matured ovum. Another, the starting point is puberty. Here the starting point is embryonic life. The process is discontinuous or staggered process. This process is a continuous process. Why oogenesis is a staggered process? Because twice it's getting arrested in cell divisional phases. Once into the embryonic life itself, when the primary oocyte started first meiotic division and got arrested at diplotiny phase of prophase 1. Secondly, the secondary oocyte, while it started second meiotic division but got arrested into the metaphase 2 stage, right? Okay. Factors needed to break the arrest and resume the cell divisions. For meiosis 1, we need two important hormones, FSH and LH. To initiate the second meiotic division, we need the entry of sperm inside the secondary oocyte. What will be the fate of first polar body and second polar body? They will not survive, they will die within few hours. Why there is unequal cytokinesis taking place? Because while primary oocyte will divide, that will form a large secondary oocyte and small first polar body. Why this kind of biasness taking place inside this process of oogenesis? Because during this long wait of 12 to 13 years, that is in between the embryonic life and the puberty, this primary oocyte will lose its one of the important cell organelle called as centrioles, and without centriole the cell cannot decide a perfect cytokinesis. That's why it will perform unequal cytokinesis, and as a result one cell will gain most of the cytoplasm and one cell will gain negligible amount of cytoplasm. So, secondary oocyte and first polar body, they are result of equal karyokinesis but unequal cytokinesis, because secondary oocyte is having haploid number of chromosome, first polar body also is having haploid number of chromosome. Primary oocyte is having diploid number of chromosome, oogonia has diploid number of chromosome. Ootid has haploid number of chromosome, second polar body also has haploid number of chromosome. So, every time meiosis 1 or meiosis 2 is taking place, cells are undergoing equal karyokinesis, equal division of nucleus but unequal division of cytoplasm or unequal cytokinesis. Yes, some Srikant, Shri, Amul, Hannasi, yes, what you people are doing here? Okay, now we'll move forward to another concept, that is the method of contraception. So, contraception means the method of prevention of pregnancy. Contraception means the method of prevention of pregnancy. Now, there are some natural methods, natural contraceptive methods, as well as some artificial methods. Now, what is the basic difference between the natural contraceptive methods and artificial contraceptive methods? Natural methods, for them you will not be finding any kind of side effect, right? But if you are going with artificial methods, for most of them one or more side effects are there. But again, if you talk about the effectivity, effective natural methods, they are not much effective, they have high chances of failures, whereas artificial methods, they are quite effective in nature. Now let us discuss some important natural methods. Okay, so as given in NCERT, first we can talk about withdrawal method or coitus interruptus. Then calendar method or periodic abstinence method. Another important method, lactational amenorrhea. So what is the importance of withdrawal method? What is the importance of periodic abstinence? What is the importance of lactational amenorrhea? Coitus interruptus or withdrawal method means when a couple is performing coitus or copulation, then the male partner has to withdraw the penis just prior to the ejaculation of semen into the female vagina. So that is the importance of coitus interruptus, or that's why it's also known as withdrawal method. But the highest chances of failure is observed into this particular method. Now let's talk about the calendar method or periodic abstinence. So in this case, we know about the menstrual cycle, and the menstrual month is about 28 days for average normal female. Now a female is knowing the dates of menstruation and what is the expecting date for the next month. So calendar wise, for menstrual month, one has to track the dates, and accordingly the couple, they can calculate the fertile period or fertile window. Now what do you mean by fertile period or fertile window? If the menstrual month is of 28 days, then you have to minus 14 with it. So for 28 minus 14, you are getting 14. So it's the 14th day that is considered as the day of ovulation. This is called ovulation calculator. Okay. Now you are not sure, you are not quite sure that is it the exact 14th day when the ovulation is gonna take place. So you need to add or minus some dates. So you have to do plus minus 2 or 3 with it. So if you minus 2-3 days from 14th day, it will be 10th day. If you add some 2-3 days, 3 days, then 2 or 3 days, it will be 17th day. So this range from 10th day to 17th day, this is the range where there is high chance of ovulation is there, right? So you need to avoid coitus or avoid mating during this particular gap, which is called as fertile window. Which is called as fertile window or fertile period. So by calculating the menstrual calendar, you have to avoid from sexual intercourse during this time. That's why it's called as periodic abstinence. To abstain means to avoid. Now what is the third method? That is lactational amenorrhea. Okay. Now lactational amenorrhea means somehow it's related to lactation or milk secretion, and amenorrhea means absence of menstruation. Okay. So if a mother is frequently breastfeeding her child, if you observe a lactating mother, lactation is happening, then there will be high titer of this particular hormone, prolactin, can be observed into mother's blood. And this prolactin is also known as lutetropic hormone, LTH. Prolactin got another name called lutetropic hormone, and that is having inhibitory capability upon gonadotropin hormones. So, prolactin will inhibit secretion of gonadotropins, means FSH and LH, and if gonadotropins are inhibited, then what will happen? Ovulation will be inhibited and menstruation will be stopped or delayed, and that is why it's called as amenorrhea, due to the frequent lactation within the first 6 months. Okay, the duration you need to mention here, this is effective only up to the 6th month, only up to the 6th month from the parturition of the baby, right. There will be high titer of prolactin present into the mother's blood, and prolactin is also acting as an anti-gonadotropin hormone, and that will inhibit the secretion of gonadotropins. And if there is low gonadotropin, there will be no ovulation. If there is no ovulation, there will be delayed or no menstruation, and that's why it got the name amenorrhea. Okay, but none of these natural or traditional methods are sure shot methods. There are high chances of failures are there. That's why you need to go for artificial methods. Now, which methods falls under artificial method? You can go with barrier methods. You can go with barrier methods. Condoms, diaphragm, valves can be used, right. You can go with pills. Options are there. Oral pills are there, vaginal pills are there, and again in oral pills there are steroidal pills are there, non-steroidal pills are there. So, there are different types of pills, they are available, right. You can go with IUDs, intrauterine devices. These are specific devices one need to install, a female will be installing inside the uterus or inside the cervix, right. Crossing the cervix inside the uterus with the help of a doctor or experienced nurse. Okay, this was quite popular among women, but not now. For the timing, like nowadays, barrier method is the most popular method, but as written in NCERT, as per the NCERT, the most popular contraception method, that is IUD, because whenever the NCERT was written, that it is the story of that time. Now you can go with injectables, or implants. Injectable and implants, there are also some other options. Okay. Now, if somebody is going with barrier method, that means you need to create a physical barrier, you need to create physical barrier in between sperm and ovum. That is why the methods are called as barrier methods. Okay. So, that will not affect any biological mechanism of the body, that is not utilizing any hormones. So, there is no side effect of barrier methods, right. So, almost it is harmless but quite effective in nature. If you talk about barriers, there are two kinds of methods they are used, that is condoms, and the second methods, utilization of diaphragms, cervical caps, and vaults. What is the basic difference between condoms and diaphragm, cervical caps and vaults? Condoms can be used by both male and female. Male condoms are available, like the example, that is okay. So, in market, male and female both kind of condoms are available. Male condom, one of the example given in NCERT, that is Nirod, a brand that is available into the government hospital, and for female a good brand, government brand, that is called Femidome, right. And most of the time the condoms are made up of the building material, that is latex. But in case of diaphragm, these are only for female. These are only for female, and that can be made either by rubber or latex. Condoms are like use and throw, they are not reusable, but diaphragm, cervical caps and valves are reusable in nature. Okay. So, SIP boosting, so what problem you are having? Okay. If you really want to watch a quality content, you can watch this. I don't know about views and all. Okay. If there is one student who is getting benefited from this lecture, then also I will be teaching. So, the basic difference between condom and diaphragm that I was discussing is that building material. Condoms are made up of mainly latex. Diaphragm, they are made up of latex as well as rubbers. Condoms are not reusable. Diaphragms, cervical caps and valves, they are reusable in nature. These can be used only by female, but condoms are available for males as well as female, right. Okay. One more added advantage or benefit of condoms are also there. It can prevent STDs, sexually transmitted disease or venereal disease. So, venereal disease or sexually transmitted disease can be prevented upon utilization or upon if you are using condoms, but this use of diaphragm, cervical caps or valves will not be giving that opportunity. So, the best option for nowadays, it is utilization of condoms. For male condoms specifically, the success rate is more than 98%, right. Okay. Now, after barrier methods, we are going towards IUDs, intrauterine devices. You need to install these. Remember, sometimes students get confused. The utilization of IUDs, where to install it? Inside vagina? No, IUDs you need to install inside uterus, but vaginal tampons, which are required after the menstruation to stop the overbleeding, to absorb the overbleeding, vaginal tampons or cervical cups. Okay, cervical cups are inserted into the cervix and vaginal tampons are inserted into the vagina, but IUDs, they are contraceptive devices inserted with the help of doctor or professional nurse inside the uterus, right. Okay. Now, different kinds of IUDs are available: first generation IUD, second generation IUD, third generation IUD. The first generation IUD, they are non-medicated. Non-medicated IUDs are there, and as you know, as NCERT is saying, the example is lips loop, and the device looks like this somehow. So, this is how lips loop is made. Notice the function of lips loop, how it works. It promotes the phagocytosis. It induces, it calls the neutrophils and macrophages into the wall, inner wall of uterus, and thus lips loop will be promoting phagocytosis of sperm directly inside the uterus, right. Okay. Now, advanced one, second generation of IUDs, they are copper releasing IUD. Copper releasing IUDs which contains specific amount of copper inside, and gradually they keep on releasing the copper ions. Why copper? Okay. Copper is spermicidal in nature. That means copper will be blocking the sperm. What is how it works? Copper, the copper ions will block the sperm from intaking the fructose. What is the source of energy for those spermatozoa? It's the fructose present into the semen, right. So, those channels by which the spermatozoa are taking those fructose will be blocked by copper. So, that is why the speed will be reduced. Speed of spermatozoa swarming or swimming will be reduced, right. So, copper ion will not be promoting phagocytosis, rather it will affect the motility of sperm. It will reduce the speed of sperm, and you know that if sperms are not vigorously moving, it will lose the capability of fertilization. So there are multiple copper releasing devices are available into the market, like copper T, copper 7, multi-load 375. Okay. What is the importance of this T7 or 375? Copper T, the device looks like a T, that's why we call it copper T. Copper 7, it's made like a 7 structured device, so copper 7. But multi-load 375, the 375 denotes the surface area of the device into cubic millimeter, sorry, surface area, sorry, square millimeter. So that's why the surface area is indicated by 375, the numerical here. But for T and 7, this is the shape of the device. Now, the most advanced IUD, they are hormone releasing IUD. Okay. Progestasert, LNG20. Okay. So this hormone releasing IUD, which hormone they are releasing? The most effective hormone which is used for contraception, that is progesterone or its derivative, synthetic derivative. Either you can use progesterone or you can use synthetic versions of progesterone like levonorgestrel. This LNG, levonorgestrel, one of the synthetic progesterone. Okay, there are few others progesterone also available in the market. Progestin, progestogens, these all are derivative, or they will act like progesterone, right? So progesterone or progesterone derivatives are released from these hormone releasing devices. Okay. Now we'll talk about oral pills. Now pills means tablet, and they are consumed, that's why they are called as oral pills. Okay. Oral pills are of two type. Oral pills are of two type. One is steroidal, means pills with steroid hormone, and another is non-steroidal. First, NCERT is talking about non-steroidal pill, and you know the name, that is Saheli. A non-steroidal pill, the chemical component that is used by Saheli, that is called as synchroman or ormeloxifene, and that is not a combination of any hormones or steroid. That's why we call it as non-steroidal. So no side effect, no steroid, no side effect. And how to use that? Once in a week pill, only one tablet in 1 week, if you are utilizing, if you are consuming Saheli, right? How it works? Will it attack the ovulation? Will it change the hormone level in female body? Nothing. Simply what it will do, it will block the estrogen receptor, and estrogen is the hormone which is necessary for the proliferation, for the division of cells into the uterus, and into that uterine bed only, uterine cells only, the implantation will take place, right? But if estrogen receptors are blocked by the chemical present in Saheli, remember that is called as synchroman, that will block the estrogen receptors present into the inner wall of uterus, and that's why it is blocking the process of implantation. It will not block ovulation, it will not affect the menstrual cycle, it will only block the process of implantation. Okay, that's why there is no side effect. But for steroidal pill, two kinds of steroidal pills are there. Pill with only progesterone These are called as mini pill. These are called as mini pill, and pills with progesterone plus estrogen, these are called as combined pills. These are called as combined pills. Okay. What is the methodology of taking steroidal pills? When we take one pill in a week for non-steroidal pill. If you are taking steroidal pill, you have to take it for 21 days continuously. Then there will be a 7 days of gap following it. So, continuously 21 days, for 21 days you need to consume 21 steroidal pills, right? And for after that you need to take a gap of 7 days, you won't be consuming those pills, and somewhere in those 7 days menstruation will take place, right? Now what is happening here? When you are consuming these pills for 21 days, the progesterone level will rise in your body. The high level or high titer of progesterone will inhibit your follicle stimulating hormone. Progesterone act as anti-FSH, and if FSH is reduced, FSH is inhibited, then there will be no ovulation, and if there is no ovulation, there is no chance of fertilization, right? So that's how it will be working, and along with that, progesterone will change the cervical quality of cervical mucus, right? If cervical mucus is thickened, if female reproductive system will start producing thick cervical mucus, then what will happen? Semen, the sperm present into the semen ejected into the vagina will not get entry passage inside the uterine cavity. Yes, slide back. Which one you want? Okay, so we are done with oral pills, and the similar kind of things, the similar mechanism of action is also exhibited by the injectables or the implants. Now what happens in case of implants? The strips, 6 strips of implants are injected beneath the skin, and gradually they keep on releasing the hormones, and the combination, the chemical composition of the hormonal composition of the implants or injectables are similar to oral pills only. Oral pills, you need to consume it orally. Implants or injectables, they are installed beneath the skin, but the durability is more in case of injectables. Like whenever you are consuming these oral pills, they will work for you, but if you are injecting those injectables or implants, they will work for you minimum 3 to 5 years. Okay. Now, if someone is looking for permanent solution, permanent contraception, non-reversal, no reversibility is needed, then they can opt for surgical method, and they can opt for surgical method. And surgical method means the method of tubectomy and vasectomy. Tubectomy means cutting of fallopian tube, vasectomy means cutting of vasa differentia. So, in case of male, vasectomy is performed, and in case of female, tubectomy is performed. And mostly they are irreversible in nature. But the surgical methods, they don't have any kind of side effects, right? How they works? Basically, everything is will be remaining normal. There will be normal secretion of hormones. There will be normal ovulation will be taking place. normal sperm production and semen production will be taking place inside the body of male. Inside the body of female, normal ovulation will take place, normal hormone secretion will take place, normal menstruation will also be taking place. They will just block the exit. They will just block the exit of the gametes. They are doing nothing. They are just blocking the exit of gametes. So if some male has performed, has undergone through this vasectomy, then what will happen? Can he produce testosterone? Yes, he can produce testosterone. Can he produce semen? Yes, he can produce semen. Can he produce seminal plasma? Yes, he can produce seminal plasma. Can he produce sperms? Yes, he can produce sperms. But while he is ejaculating the semen will not be containing the sperm, rather it's only containing the liquid secreted by those seminal vesicles, prostate and other things. Okay. So that was about the contraceptive method. Now if we talk about infertility, we need to take help of art. Now what is art? Art assisted reproductive technologies, right? Now help of art is taken when doctor declares the couple or one of the partner of the couple is infertile. What is the definition of infertility? A couple is cohabitating together, and for how many years? For 2 years. Upon 2 years of continuous coitus or cohabitation, sexual intercourse, after that also they are unable to conceive a baby. The mother is unable to conceive a baby. Then the doctor will advise to take help of assisted reproductive technologies. Now assisted reproductive technologies, they are of two types. One is called as IVF, in vitro fertilization, and another is in vivo fertilization. In vitro fertilization means fertilization outside the mother's womb, fertilization inside the laboratory. In vivo fertilization means fertilization inside the fallopian tube of mother. So another terminology that is used for IVF, that is test tube baby. But you need to remember that often we use the term test tube baby program to talk about when we are talking about IVF, but literally there is no use of test tube in laboratory. Rather, highly sophisticated tissue culture, animal tissue culture techniques are used. Okay. So for test tube baby or in vitro fertilization, in real time there will be no use of test tube, rather some other tissue culture techniques will be used. Okay. So it's for layman people. Layman will not understand what kind of techniques we are utilizing in animal tissue culture laboratory. So to make them understand in a similar or easier way, they have explained this in vitro fertilization method by saying it's just test tube baby formation, right? Okay. Now in vitro fertilization is having ET techniques, embryo transfer technique, where one can go for ZIFT and intrauterine transfer. IVF contains ICSI technique as well. Now what are the difference between embryo? Okay, what is the difference between ZIFT and IUT? Both of them are embryo transfer technique. Sperm and ovum taken out from the parents, right? Either it can be the biological father or mother, or the gametes can be taken from the donors. Okay. Now under special condition in laboratory, fertilization will be stimulated, and after fertilization there will be formation of zygote. After zygote there will be formation of morula, right? Now if you are replacing a embryonic stage which is less than 8 cell stage, less than 8 blastomere stage inside the uterus of mother or surrogate mother, and that that process is called as ZIFT, zygote intrafallopian transfer, right. So, what is happening here? The cultured embryo which is less than 8 blastomere stage, the embryo with less than 8 blastomere stage will inserted into the fallopian tube of the biological mother or the fallopian tube of surrogate mother, right. Okay. Now, intrauterine transfer, what do you mean by that? If you are transferring more than 8 blastomere stage inside the uterus of the biological mother or surrogate mother, then that process is called as intrauterine transfer. Okay. Someone is saying, sir, please explain MTP. Okay. MTP is a different thing, that is called medical termination of pregnancy. For medical termination of pregnancy, that's not a contraceptive method. That is bounded by some legal terminologies, right. If female is pregnant due to rape case or unwanted pregnancy is there, the contraceptive methods has failed, or the child is with abnormalities, or the mother is with physical or mental agony, then in that case under certain medical ground, under the observation of 1 or 2 doctors, within first trimester the embryo, the fetus can be aborted. So, that is called as medical termination of pregnancy, and that is quite safe if someone is performing within first trimester. If you are going for second trimester, then it will be much riskier, right. So, within first trimester, one observation of one medical practitioner or one doctor is enough, and if you are going beyond that, you need to take consultation from 2 doctors at least. So, that is simple thing about MTP, and in a year near about 45 to 50 millions of MTPs are performed, right, throughout the world, and which is near about 1/5 of total pregnancies which are caused. Now, again another very important method of in vitro fertilization, that is ICSI, intracytoplasmic sperm injection. So, what happens here? Secondary oocyte is taken from the mother, and under regulated condition in laboratory, the sperms are inserted or injected into the cytoplasm of this oocyte. Now, what will happen in natural way? There will be fertilization taking place inside these inside the laboratory, inside the tissue culture facility, and formation of embryo will be there, and that embryo will be transferred by following ZIFT or IUT. So, when sperms are directly injected into the cytoplasm of the oocyte, then that process is called as ICSI. For ZIFT or IUT, naturally the fertilization is taking place inside the tissue culture facilities, but the sperm injection is not done here. Okay. Now, when to perform ICSI? Suppose there is a male who can produce sperm, but those sperms are non-motile. Sperms do not have the capability for swimming. So, in that case, ICSI can be performed. For in vivo fertilization, generally we are going with the gamete transfer technique. Generally, we are going with gamete transfer technique instead of going for embryo transfer. Now, for male, okay, if you are taking gamete from male, then by the process of artificial insemination or intrauterine insemination, the semen is injected into the vagina of female. If you are taking gamete from female, then the technique that is performed, that is gift, gamete intrafallopian transfer. Do not get confused with zift and gift. Gift is a in vivo fertilization technique. Zift is in vitro fertilization technique. In case of gift, a functional ovum is collected from a healthy female and inserted into the fallopian tube of the biological mother or the surrogate mother. Generally, gift is performed in that case, in such case, where the mother, for a couple, if the female partner or the mother is unable to produce the ovum, the condition is called as anovulation, but can provide healthy environment for implantation and the embryonic development. So, in that case, ovum will be taken from another donor female, and that ovum or the secondary oocyte will be inserted into the fallopian tube of this uh, female partner of that particular couple, and gift will be performed. Okay. Someone is asking, okay, Ruhi Fatima, where from you are? Ruhi Fatima, I just wanted to know that from where you are watching this. I'll explain that. The steps of urine formation I will explain, but from where you are watching. Yes, Pranika. Uh, Harsh, no, that's not, this series is not for typically for NEET preparation. It's actually, it's a kind of bridge course, means very basic explanation, very basic discussion. Those who are coming from class 11 to class 12, this is particularly for them. So, a very light touch to them, what they are gonna study while they will be coming into class 12, right? Obviously, these lectures are not in depth or in detail in classroom. Whenever we will be studying, then the lectures will be given in much uh, detailed manner. More diagrams will be there, question practice will be there, uh, explanation of each and every lines of NCERT with proper format will be given to you. This is just for a kind of a mock drill, right? Khazana, I don't know which batch you are taking, which batch, Pranika. So that should, I think that is available. Okay, so Khazana, I don't know about Khazana, I have to check for that. But yes, after every lecture in PW app, you will be getting the uh, top, like top most important question for that particular topic or for that particular chapter, so that you can practice as a DPP, daily practice sheets, right? So, if you are practicing all the questions which are discussed in the class and given into the DPP format after every class into the app, so those things will be enough for you. Yes, Sachin Singh, you are from SS Public. Okay, where it is situated? Is it in Bihar? Okay, any more question from anybody? Sachin saying, sir, I have learning from you, sir, at SS Public. Okay, I got it, Sachin Singh. Yes, I can recall it, though it's near about many years, 6, 7 or 8, 9 years. So how are you, Sachin? How you are doing in your life? Okay, any question? Any one of you want to ask? Sir, how to read NCERT biology? Great. That's a good question. See, NCERT is the heart of NEET preparation. So, in the beginning, when you were getting NCERT and you started reading it, will find it quite boring. Pranika, I'm telling to you. In the beginning, you will find that NCERT is quite boring. But when do you have to read NCERT actually? First, listen all the lectures from your teacher. Like if you are enrolling in our batch, that is Laksha NEET and Laksha NEET English, right? Whenever we'll be teaching in a daily basis or weekly basis, after every class, you have to check the topics or check those concepts from NCERT. Line by line, you have to read it, and you have to you will try to recall what my teacher told during the time of class. Or is it syncing? Is it synchronizing? And gradually, what you have to do while you are while you have started reading those lines or paragraphs of NCERT, you have to start framing your own questions. You have to make your own questions from your inside. So that will give you that positive mentality, so that whenever you will be appearing for NEET 2027, right, uh, the examiner mentality will be inculcated inside your mind. So first you have to read the introduction. Prior to every section, every unit, you will be finding introduction. So most of the students, they think that is irrelevant. First you have to check that introduction session given prior to each unit. Then you will get the chapter. Then you will get the chapter. And what is present inside the chapter? Line by line text is written in the chapter. Once the chapter is completed, you will be finding a summary section. Once you are done with the text, with the summary, after every chapter you will get exercise. This exercise part is not only important for those who are preparing for board, but also MCQ can be asked from exercise, and you will be finding 1-2 questions those are not mentioned into the text itself. So that's why from top to bottom inside NCERT you have to read each and everything, right? And whenever you are reading by your own, obviously you will be finding it it's quite boring for you. So that you have to take help of the lectures of your teacher, and side by side your teachers has given the lecture on upon a particular concept, and after completion of that you have to go for reading of your NCERT and figuring out your own questions along with practicing the daily practice sheets. Oh, here I got Hamza Ali Mazari directly from Liari. So, how's going on, Mr. Hamza Ali Mazari from Liari? Yeah, for biology, only NCERT is enough. Okay. Max to max, you can get 2 questions to 3 questions out of NCERT. Max to max. In worst case, I'm telling it, in worst case, in case of NEET examination, for biology particularly, for physics, you have to go out of NCERT, for chemistry as well. But for biology, max to max 2 to 3 questions can be expected in coming years. But 2 question means only 8 marks. To get a decent rank in NEET examination, you have to score 340 or 350. Okay, so if you are skipping those 2 questions, so uh, for those 8 marks, you cannot disturb your whole schedule by reading all the thick books which are available in the market. NCERT along with your like the study modules, whatever you are getting, along with practicing questions and listening the classes, more than enough for biology. Sir, my biology is good, but I am not confident. Yes, that's the case for almost 90% of the students. So what happens? You feel that you are good at biology because somehow you can recall the concepts, but you are not reading the NCERT in an insightful manner. Reading the NCERT and reading the NCERT in an insightful manner, these two are totally different thing, right? So for that you have to read the NCERT in an insightful manner. NCERT is a very boring kind of book for a student perspective, right? But when you understand the concepts and then try to find out questions inside the lines of NCERT, then it will appear interesting to you. Can we crack NEET preparation from class 12? Yes, if I don't know about the base of your class 11. If already you have done quite good in class 11, then obviously if you are uh, taking the Lakshya NEET English batch, obviously you will be finding fantastic faculties with conceptual clarity for every classes. After every classes you are going to get good, like not only questions but relevant questions after every class as DPP, daily practice paper. And along with that, if suppose you have some backlogs, so whenever you will be getting some times, I think you will be getting some time, because for class 12 the whole and soul syllabus, it will take maximum 5 months to get completed, right? In compared to class 11 syllabus, biology syllabus of class 12, it's very precise. So quickly we need to complete the class 12 chapters, and after that you have to start revising the chapters for class 11 as well, and obviously your faculties or teachers are there to help you to recover your backlogs. So why not? So I just assume that my bio is good, but when it comes to reality, I cannot score good. Yet it happens. Biology will make a resistant, a score like 280, 300 for most of the students. Those who feels that they are good at biology. So to break the resistance, what you have to do for that, you can watch the strategy videos which are available in PW NEET English channel. There is a channel. Uh, this is PW English medium. Okay, there is a channel called PW English medium. You can check the strategy videos, and 2-3 videos you will be getting which is similar. I was trying to explain the strategy, how to crack the resistance or the barrier to cross 320 or 330 or 340. So that's what we call as overconfidence. No, you need that confidence. You need that confidence, but you need to channelize in a proper manner. Yeah, you can use any study material for question practice. All questions are good. Sachin is saying, sir, I am also preparing for NEET last from 2 months, and I am following your free YouTube series. That was the great helpful for me before that. Great to hear that. When our efforts are helpful for our students, so at the end of the day, we can sleep peacefully. Okay, then for today, it's bye bye and good night. Jai Hind. Okay, again Hamza is saying sometimes I feel like we're just having 1 month left and I'll not be able to read NCERT twice. No, it's not like that. take deep breath. You have 25 days in front of you. You have nights, you have days, and each day counts 24 hours. Sleep well, and apart from that, invest your energy, your focus, your all the motivation behind all these things. Don't worry about that. You have enough time. Okay, you have to control the motivation. It's you who can motivate you at the highest manner. Okay, the external motivation, those are false, those are fake. You have to enlight the internal motivation. You have your time. Every day counts. Every hour counts. So you have to utilize them in a proper way. Okay, like your name Hamza Ali. Okay, if you have watched that movie, so you have to do comeback. Don't worry. Good. Okay, good night. Pranika. Okay, my name is Biswajit. Welcome, welcome Hamza. Thank you so much, sir, for encouraging us. I must say you are one of the best teachers. Okay, it's my pleasure. It's my pleasure. I'm not best, but always I try to teach my students from my heart. Yeah, surely we have to do comeback. We have to overcome every barriers of our life. Success is waiting for everyone. Don't worry.